So, good morning, good afternoon, or good evening, depending from where you are joining us. Tonight, the IVPN NeuroPsychiatry, supported by Pfizer, is organizing the first Migraine Masterclass, our one-of-a-kind learning experience. During this educational activity, our top-notch migraine experts will discuss with you the management of migraine in the 21st century. What are the latest findings in the genetics of migraine disease, and what are the new treatment approaches? What is special about this masterclass that they will be endorsed by the American Association of Migraine Disorders. And this endorsement wouldn't have been possible without the great effort of Dr. Hiba Bani. Let me introduce for you our moderator, Dr. Hiba. So Hiba, she's the founder of the Migraine Pharmacist. She's the program manager of the Migraine Pharmacy Network in Canada. She's a registered pharmacist with an MBA in healthcare and life science. She started her career in the pharmaceutical industry, managing migraine brands such as Imitrex and Botox, and she currently trains pharmacists on managing migraine throughout the MPN program. She has been counseling migraine patients in person and virtually in Canada as part of WellCA, the leading Canadian health and wellness provider. Dr. Hiba works closely with the Migraine Canada and the Canadian Migraine Support Group, among other organizations that support people living with migraine. And also she is a board member for the American Migraine Association, the Association for Migraine Disorder. So welcome Hiba to this educational activity, and let me thank you again for all the effort and for endorsing our activity. Please, the floor is yours.
Thank you very much. It's a pleasure to be here. Thank you very much, IVPN, for giving us the opportunity to work together and bring such an amazing program to the whole world. This program is going to be available in the Middle East, North Africa, North America, and anybody who is interested in learning more about migraine. Just to give you a quick introduction about who the Association of Migraine Disorders are. This is an organization that is based in the US that strives to expand the understanding of migraine by supporting research, education, and awareness. Supporting research is through providing seed funding that stimulates collaborative networking and cultivating a new generation of experts. Education, we are today in one of these educational efforts where we deliver easy, accessible education to providers and to patients, and awareness, mainly working with social media and direct advocacy for people living with migraine. It gives me great pleasure today to be joined by two of the experts in this area, and it's such a pleasure to have Dr. Lynn Griffiths. Dr. Lynn is joining us from Australia, so I think it's almost 1 a.m. for her. This is unbelievably generous from your side and dedicated from your side, Professor Griffiths, and we're really, really happy to have you here. We will start today with Dr. Lynn Griffiths, and then for the first part, and questions will be after that, and then we will have Dr. Tawfiq Saadi from the UAE, and who will be taking the second part and answering his questions after, or questions directed to him after his session on the second part. Please put all your questions at the in the question and answer section of this event. We welcome you all, and we hope that you have a very informative presentation or session today. To go back to Professor Griffiths, distinguished Professor Griffiths is an active and respected molecular geneticist. So we wanted to start this session with genetics, to start right at the beginning. She has more than 30 years experience, and she has brought a translational focus to medical research to increase the influence of her practice and her research and the impact of Genetics on Human Health as director of the Center for Genomics and Personalized Health, which is focused on translating knowledge from genomics research. The center aims to discover better methods for diagnosing disease, develop targeted treatments based on genetic information, and train the next generation of translational genomic scientists. In addition, she is a passionate advocate for the translational for the translation of medical research through commercialization, and is currently the director of the MTP Connect and industry-led Bridge and Bridge Tech programs, undertaking commercialization training for the pharmaceutical and medical devices technology fields across Australia. Dr. Griffiths owns genetics research at the Genomics Research Center. Her own genetics research at the Genomics Research Center has led to diagnostic breakthroughs for several neurogenetic disorders, including familial migraine, ataxia, epilepsy, and hereditary stroke. Her research has appeared in more than 400 peer-reviewed international journals, and she has obtained significant competitive and industry research funds to support her research team. Without further ado, welcome, Professor Griffiths.
Thank you so much, Hiba. And it really is very late here. It's not 1 o'clock in the morning, it's after 2 o'clock in the morning here. So it's a very strange time. So I'm going to try and share my screen, and let's hope it all works well. So just want to check that everybody can see that and that it looks reasonable. So first of all, I just want to thank you very much for the opportunity to present to this first migraine masterclass, and particularly with a focus on genetics of migraine, an area I've been interested in for many years. So I'm just going to start off with just a little bit of background. I'm then going to talk about what we know about migraine genetics and the ways that we can approach trying to identify the genes that play a role in migraine. But I'm also going to look at what we currently can do with diagnostics and think about some future things. So as we're all aware, migraine is a very common painful disorder that affects a significant proportion of the population. There are no simple laboratory-based diagnostics for the common forms of migraine, although there are some genetic tests available for rare and monogenic forms of migraine. As I mentioned, migraine is an extremely common disorder affecting a significant proportion of the population. And it doesn't really matter what population you look at. There's been quite a number of studies across different populations around the world. And in just about all cases, you'll find that migraine occurs much more often in females than males. And it's usually about 3 times more common in females than males. You can actually see in terms of age of onset that there are different ages when people can develop migraine, but you'll find for males it varies from quite a young age, but you'll notice in females this big peak that occurs around about puberty for the age of onset in females. So this clearly indicates that this is the increase when you start to see females being affected so much more by migraine. The other thing to note about migraine is that it does show comorbidity with a number of other conditions, including cardiovascular disorders such as stroke, epileptic disorders, diabetes, and also psychiatric disorders. So it is possible that there could be shared genetic factors in there, and this could provide us a bit of a hint in terms of what we should be looking at. In terms of migraine, we know what the symptoms are in terms of how we would diagnose it. And we also have been able to define the 2 types of migraine, with and without aura, now a lot more clearly. I think the thing about genetics, though, which is particularly interesting, is that migraine does show strong familial aggregation, meaning that it runs through families. And about 90% of migraine sufferers have a close relative who also suffers from the disorder. Twin heritability estimates vary depending on which particular population you're looking at, but it's usually around about 60 to 65%. So meaning there's a significant genetic component, but there are also some environmental factors and components that play a role here. So migraine is a complex disorder. It does have environmental components as well as that significant genetic background. To make it complex too, there are the two main types of migraine, and it was thought originally that maybe there would be different genes for the different types, but it is fairly complex because you'll find both migraine with and without aura run through families, the same families, and even the same individual can suffer from different types of migraine at different stages. As I've mentioned, there's also the comorbidity of migraine with other disorders. And in terms of genetics, when we first started working on migraine genetics, and I certainly started quite some time ago, we were kind of hoping that there might be just a number of key genes involved in the disorder, but we now know that there's a very significant number of genes, and we certainly haven't identified all of those. So at present, the number of genes is unknown. Rare and severe forms of migraine, though, and certainly for common migraine, there's probably many genes, but for the more rare and severe forms of migraine, we do know that there are some monogenic forms, meaning that there'd be a single gene that plays a role in specific families and a causative role. So how do you actually go about trying to identify the genes that play a role in a disorder? Well, there's two main ways to do that. So one of them is to look at case control populations, and the cases and the controls need to be carefully matched in terms of things like age, sex, but also ethnicity, so that you're not picking up genetic variants that might be playing a role in ethnicity or that might be playing a role in the gender differences. So that's called a case control approach. Second approach that's often used to investigate genes for a particular disorder is to use a family-based approach. And migraine is a disorder that clearly runs through families. So this is one of our migraine families, and you can see that it's cropping up in every generation. Occasionally it might be missing a generation here. But in general, that could be because it may not be as severe in the person, so it may not be diagnosed in that person, or it could be really that they're just not manifesting at that particular stage of life and may have manifested at other stages, perhaps not even diagnosed. So it's quite easy to actually get quite large families like these who suffer from migraine, and that way you can then start to track genes that might be inherited or segregating with the disease from one generation to the next. The third way of looking, first is case control, the second way is to look at families, but the third way is to look at what's called founder effect populations. We've actually been working with one from an island off the coast of Australia called Norfolk Island. Norfolk Island is what's called an isolated founder effect population. And in fact, the majority of the people on the island can trace their ancestry back to a kind of famous historical event, and that was the mutiny on the Bounty. So there was a mutiny on a ship that was traveling a long time ago in the 1700s. It's called the Bounty, and it was traveling from England through the Polynesian islands and was aiming originally to get to the West Indies. Stopped around Tahiti. There was a mutiny then, and a number of captain and people were put off the boat. But the long and short of it was that Fletcher Christian, who led the mutiny, and his supporters took the boat with a number of Tahitian women that they were happy to travel with. And all of this population then went across to a small island called Pitcairn Island, and proceeded to be there for many, many years. They had children and they grew quite comfortably in terms of size. Rediscovered in the 1850s. Queen Victoria then pardoned the offspring and moved them all across to the island that I first mentioned, which was Norfolk Island. Norfolk was an uninhabited island. So basically, people from the 1780s, 12 maternal Tahitian women, 9 paternal Bounty mutineers, were the founders of the majority of the current population on Norfolk Island. So we've been working with that population since the year 2000 to undertake genetic studies. We've put together a big pedigree, and this is just a small component of the pedigree. We actually have a pedigree with about 6,500 people in it that goes back 12 generations to the original founders, and we've collected samples over about 5 generations, and we've collected them longitudinally since the year 2000, 2010, and more recently now in 2021. We've undertaken a lot of DNA studies, but also phenotype studies and investigated them for CVD risk traits, ocular phenotypes, but also migraine. Interestingly, when you look at that population, we found that there's quite a significant prevalence of migraine. About 25.5% of the population suffer from migraine on the island, which is a lot higher than what you'd expect in Australia and New Zealand, which is thought to be around about probably 12 to 15%. It also shows significant heritability, meaning that there's a number of genes that are playing a role in that population on Norfolk. And this is exactly the sort of thing that you would find in an isolated founder effect population. If there are genes for disorders, and it's kept within a closed population, you will tend to find that amplified. So there would be more people affected as it's kept in the population. So this is the third way that we've currently been investigating disorders, and this is a way that a lot of other people have also been investigating, trying to find genes in disorders. So if we actually start, I just want to see if I can shut this, that's better. So if we actually start looking at how you can then start to identify genes involved in a disorder like migraine, there's two types of variants that generally play a role and that tend to be investigated when you're looking at genetics of a disorder. The first are called rare variants. So these are less than 1% in terms of their minor allele frequency. Many of these rare variants are benign, but however, if they do occur within a disease within a gene, and they change something in that gene, such as an amino acid, and change the function of the protein or the effect of it in terms of gene expression, then they're no longer benign. They may then be pathogenic, and then they can be classified as a mutation. So rare variants can be mutations that are pathogenic and can cause disorders. The other type of variations that can be investigated are common variants, and common variants in the opposite way have a minor allele frequency that's greater than 1%. And most of the common variations that tend to be used for genetic studies are what's called single nucleotide polymorphisms. So that's a variation at one point within the genome where one of the nucleotides is changed from one base to another base. There are many of these. There's approximately 10 million in the human genome, and it's believed that there's about one SNP every 300 base pairs. These common variants can also influence traits and also influence disease risk. So both of these can be used for studies. And so when we look at the sorts of studies that you can undertake, I mentioned before that people tend to use genetic association studies when they compare cases and controls. And the sorts of markers that are often used for these are those SNPs, single nucleotide polymorphisms, because they can act as biological markers to help locate the genes that are playing a role in a disease or a trait. It could be that they play a direct role. If it's a gene, it's a variation that occurs within the regulatory sequences of a gene, or it could be that they're actually co-inherited with a causative variant. So they could mark the genomic region that's associated with the trait. So SNPs are very commonly used now as genetic variants or markers to investigate disease. And there's probably two ways that this can occur. And a lot of the earlier studies focused on what was called candidate gene association studies. So this means it's investigating variations that occur within specific genes that could plausibly play a role in the disorder, that is candidate genes. And for migraine, there are many candidate gene studies focusing on things like neuronal genes, vascular pathway genes, or genes that play a role in hormonal pathways. Instead of investigating just specific SNPs within particular genes, though, a more comprehensive approach is to use a genome-wide association study. And what this does is investigate SNPs not just in one gene, but across the whole genome. And that way you can perform association tests for many markers across the genome. Because you're testing many different markers, and many of the arrays that have been used for SNP testing now have up to about 1 million SNP markers on one of those arrays. So because you're testing so many arrays, it's really important to correct for multiple testing but also to have sufficient power to detect something significant, it's important to have large sample sizes to investigate these for power. So in terms of studies that have been undertaken, both candidate gene and GWAS studies have been used to investigate migraine genes. There's been quite a number of specific genes that have been implicated, and some of the ones listed up the top here, such as estrogen receptor 1 and 2, progesterone receptor, and some of the others, such as MTHFR, have been investigated as candidate genes and shown association with either migraine with or without aura, or with migraine in general. There's others that have been investigated using the genome-wide association approach, and these have involved much larger migraine populations, and more recently have involved the International Human Genetics the International Headache Genetics Consortium, where a number of groups across the world have pooled their samples together to make sure that we have sufficient power to investigate appropriate numbers of cases and controls and detect significant results. And these have implicated also a significant number of genes. Other approaches, as I mentioned, have utilized families, and one particular gene, the TRES potassium channel gene, has been implicated using a large migraine with aura family. And this has shown complete segregation of a frameshift mutation through that family. And then finally, other approaches have investigated the more rare but more severe monogenic forms of migraine, and that has implicated a number of specific ion channel genes that I'll talk a little bit more about shortly. So as I've mentioned, how to identify those has involved genome-wide association studies, and this is one of the earlier ones. Also, large migraine family and DNA sequencing implicating a frameshift variant within the TRES potassium channel gene, but also genome-wide association studies. And these are the results of one in 2016 that identified 38 different susceptibility gene loci for migraine. And the very recent one that's come out now in 2022, again, it's a collaboration of the International Headache Genetics Consortium, has shown now 123 risk loci for migraine. So certainly as we increase the power and the number of cases and controls that we can investigate, the more we are detecting a number of genes that are involved in these common types of migraine. And if you look at those genes, and there's many, as I've mentioned, most of them tend to fit within either those that have neuronal or vascular function. So if you look at this diagram, this outlines some of the ones that have been implicated. Those outlined in black, such as these ones, are from genome-wide association studies of the common types of migraine. And those outlined in red tend to be using families investigating monogenic forms of migraine where there are single causative markers within specific families. If you look at those that were identified using association studies, such as GWAS studies, most of those associated SNPs have relatively small effect sizes, with odds ratios of about 0.9 to 1.1. However, you can start to pool some of those associated SNPs together to give a profile and what's called a polygenic risk score. And this provides a good estimate of the effect of those genes on migraine. And what it does show is that we can identify a profile for migraine, but more importantly, what it does show is that it still doesn't explain all of the heritability that plays a role in the common forms of migraine. So we do need to start thinking about how we can take that forward to explain all of that heritability. Possibilities, again, are to increase the sample sizes to make sure we have well-characterized case and control populations. But perhaps we also need to be thinking about other types of genetic markers that could be playing a role in migraine. So these could be epigenetic markers, which is not so much changes in the DNA, but things that affect the expression. And I'll come up to that a little bit later as well. But the other one is perhaps there are not just common variants, but maybe rare variants that are also playing a role in the common types of migraine. So this is obviously an area that's ongoing research in terms of migraine with and without aura. And it might take us a little while to actually start to increase those numbers. But as I've said, there's now the most recent one has identified 123, and there will undoubtedly be more markers involved in migraine. So right across the genome, we'll continue to find markers, but I want to focus a little bit more on those monogenic forms of migraine at the moment. So when we think about those, there are three well-known genes that play a role in what's termed familial hemiplegic migraine. So there are the three corresponding types of this. So migraine, familial hemiplegic migraine types 1, 2, and 3. This is a subtype of migraine with aura. When it runs through families, there are some sporadic forms, but the majority do tend to be familial. It shows autosomal dominant inheritance and also a relatively early age of onset. So in addition to sensory disturbances, FHM manifests with some more severe symptoms, including things like hemiparesis, deafness, stigmas, also effects on the retina, retinal degeneration, and in some cases, even coma. It has been linked causally to mutations within three specific genes. So the CACNA1A gene, ATP1A2, and the SCN1A gene. So if we think about the first gene, and this one was the one that was first identified a number of years ago by Opper et al., CACNA1A we now know has quite a number of mutations across the whole gene, and it's different mutations in different families that play a causative role in FHM1. So these are gain-of-function mutations. They're inherited in an autosomal dominant fashion, and they result in individuals, quite often, as I've said, from a young age, suffering from FHM1. Interestingly, most of these are missense mutations, so single point mutations, a change of a single base, and as I've mentioned, they're usually gain of function. However, interestingly, there's also additional phenotypes that are due to mutations within the same gene. So episodic ataxia type 2 is also a genetic disorder. This one's primarily characterized by episodes of poor balance and coordination, and it usually involves not so much gain of function, but loss of function mutations in the same gene. Episodes can be precipitated by some specific factors such as trauma, stress, or particular chemical triggers, and additional symptoms include partial paralysis, altered vision, slurred speaking, etc. There's also often an effective treatment in the form of acetazolamide. So here's two different disorders coming from the same gene with different types of mutations. And then another third phenotype, again from the same gene, is spinocerebellar ataxia type 6 or SCAR6. Sufferers from this show poor coordination, speech problems, some vibration and other movement problems, and sometimes cerebral atrophy as well. This is due to a totally different type of mutation, and the most common type of mutation is a CAG triplet repeat expansion that can lead to an extra long polyglutamate region in the 3' end of the gene. So normally it's between 4 and 18, and those affected, it goes up to 20 to 33. So that was FHM1 gene, the CACNA1A. So similarly, the second FHM gene, FHM2, this was identified originally a number of years ago in a number of Italian families that showed inheritance or segregation of specific mutations within the ATP1A2 and complete segregation with FHM within those families. So this one has been termed the FHM2 gene. So as I've mentioned, it's causative of this particular type of FHM, but it's also been associated with another phenotype, this one with alternating hemiplegia of childhood. This typically develops in a really very young age of 18 months of age, includes episodes of paralysis, maybe hemiplegic, or it can affect different combinations of limbs. And episodes can last hours to days and sometimes can resolve after sleep. Also, walking balance problems can occur as well, and it can sometimes be accompanied by seizures and or developmental delay. So again, two phenotypes with the same specific gene, but with different mutation points. And then the third FHM gene is the sodium channel gene, SCN1A, and this was first identified as causing FHM3. But interestingly, again, there's another phenotype that potentially plays a role with this one. So, and this relates to epilepsy. So as mentioned before, there's some comorbidity between migraine and epilepsy, with epilepsy patients with two or more first degree affected relatives having a twofold increase in the risk of having migraine with aura. There's also a syndrome of migraine with aura immediately followed by an epileptic seizure. But most importantly, and going back to this specific gene, SCN1A, we know that specific mutations in this gene can cause FHM, and this one's classified as type 3, but also mutations in the gene can be causative of Dravet syndrome as well, which is a common pediatric epilepsy. So it's interesting when you start looking at FHM genes and the symptoms associated with them. So you can look at the CACNA gene and outline of the symptoms here, but mutations within this gene can cause not only FHM, but also episodic ataxia type 2 and spinocerebellar ataxia type 6. ATP1A2, if you look at those symptoms, that can occur in those seizures, loss of consciousness, and mutations also occurring in alternating hemiplegia of childhood. And as I've mentioned, in SCN1A, number of symptoms here, and it can relate to, mutations can relate to FHM3, but also Dravet syndrome. So despite these associations with the genes and with the symptoms, there's actually considerable variability between and within families. And also in terms of diagnostics and doing things like sequencing, sometimes it can be difficult based on symptoms to determine which particular gene might be causatively involved. And to make matters even worse, there's another disorder called CADASIL. This is also a dominantly inherited disorder, but it's one of small vessels of the brain. This one occurs a bit later, usually fourth or fifth decade, can lead to cognitive impairment and also dementia. And it is the most common form of small vessel disorders. So some of the symptoms associated with this would be migraine, stroke, but also white matter lesions. And it's usually migraine that is the first presentation of the disorder, and this can occur much younger than the cognitive symptoms. So it's a fairly complex aura. It's also can be combined with early stroke episodes. And as I mentioned, some of this tends to overlap some of the FHM symptoms and vice versa. Interestingly enough, the gene that plays a big role in CADASIL has been identified. This is the Notch3 gene, and Notch3 plays a causative role with single mutations within this gene being inherited in that autosomal dominant manner through families. So if we start looking at FHM, it's a disorder that has a prevalence of somewhere between 1 and 10,000. As I've mentioned, the symptoms associated with it can be variable. So the first one was the most common ones, but there's also 15% of people show alternating FHM and migraine with aura attacks. Quite a significant number show prolonged aura attacks. 25% show bilateral sensomotor disturbances. 20% of them show episodic, and also some of them show some associated seizures. There are a number of treatments for FHM as well that can relate to some of those and be helpful for individuals. If we start thinking about some of the symptoms associated with episodic ataxia, so again, that same gene, you can look at that and you can see that there's definitely overlapping symptoms between FHM1 and episodic ataxia. Also, if you start looking at CADASIL, again, some of those symptoms are overlapping, but all of these have different genes involved and also potentially different treatments, particularly for things like CADASIL. So why is that important? Why is it important to be looking at monogenic forms of migraine? Well, certainly it's important for diagnostic purposes so that you can determine which particular disorder you're looking at. Also, as I mentioned, there may be different treatments for different types. And also, it just makes it easier, I suppose, to differentiate whether you're looking at migraine or other disorders as well. So genetic testing is now available for FHM and related disorders. And in fact, our lab's been involved in that for many years. We've been an accredited diagnostics lab since 1999, started off looking at the CACNA1A gene, have extended diagnostics to include other genes as they've been identified. And we currently have been able to undertake tests and do tests for all of the disorders that I've mentioned here. In Australia, the accreditation system is what's called NATA accreditation, and we've had that accreditation since 1999, and we grow that from our research interest originally, and we also change our technology as things tend to change. So originally, we used a method called Sanger sequencing, which has always been in the past the gold standard for DNA sequencing. It's very reliable, but slow and expensive. And in 2013, we switched over to a newer method called next generation sequencing. We were, in fact, the first to set up a next generation sequencing accredited panel for these target disorders. And that's been accredited, was set up in 2012, but accredited in 2013. So just thinking about that, since we do all of the diagnostic tests for Australia and New Zealand, we have had quite a number of samples over the years sent to us through hospitals, through pathology labs, and through neurologists to investigate potential patients with these disorders. So when we did the Sanger sequencing method, we had quite low diagnostics rates. So just looking at the 700 patients that have been sent for us for diagnosis, we were able to detect only about 8% with identifiable mutations. The methods are also, as I mentioned, time-consuming and expensive. Most people would have picked a specific gene. We tend to focus on the exons with most of the mutations that are known to be there. We then might go out to other exons. We might then move on to, if that gene was negative, going to the next gene. That made things slow, but it also made things expensive too, because the method was expensive. So what we did was we set up a method allowing us to investigate all of those genes at once, and that's using a next generation method. Doing that, we covered about 92% of all of those genes, and we were able to then test that on the samples that we had, so we could verify and validate that it was a correct method and get it accredited, but we could also determine whether it actually helped in enabling diagnosis. So as I mentioned, we had about 8% method when we used the Sanger older methods, but when we switched to next generation sequencing, we got it up to 32% of FHM patients and about 20% of CADASIL patients. So overall, there was an increase of about 7.7 to 26%, so about a fourfold increase in mutation rate. Interestingly, some of the patients that were sent to us, here's an example of one with a potential episodic ataxia. So we were asked to test for mutations in the CACNA gene, but because we're now using a panel that covers all those genes, we actually detected a mutation in the ATP1A2 gene, that is FHM2 gene. Another couple of examples for case 2, this is one that was referred for Notch3 screening for CADASIL, but it actually was a mutation in the CACNA1A gene, so doesn't have CADASIL, actually has FHM. And then the next one here were 2 brothers referred for FHM screening, but they actually had mutations in the Notch3 gene, which are causative of CADASIL. So interestingly here, you can find that using a panel approach, it costs a lot less. It's about a quarter of the previous costs. No need to do iterative testing. It's much faster. And there's also no need to define which specific gene that you're interested in. Even so, though, we're still only getting about 1/3 of the cases diagnosed. So clearly there are mutations that we're missing. We've now recently developed another panel with 15 genes. We've brought in some other genes that have been implicated in FHM, such as the PRRT2 gene, which has now got some accumulated multiple reports of FHM symptoms in mutation carriers. A couple of other genes that have been causative in a number of reports for ataxias. ATP1A3, which can also cause alternating hemiplegia of childhood. And we've also introduced some other genes that play a role in small vessel disorders, such as HDR1 for carousel, COL4A1 and 4A2, which has also played a role in similar stroke-related disorders. A couple of others that also play a role in similar stroke-related phenotypes as well. So we now have a panel that covers 15 genes. It's got a coverage of 99.19%. And it's also been accredited as a diagnostic service. So that's a good way of, if you think that you're looking at FHM or you're looking potentially at CADASIL or one of the ataxias, that's a good way of targeting and looking at those genes. But what if it's a more complex phenotype, such as epilepsy, or you get a negative result out of the panel? Well, we've also recently accredited a whole exome. That means that you test all different exons across the whole genome. And so using that approach, you actually pick an awful lot more. When you start looking at all genes across the genome, you're going to get a lot more variants that you have to investigate. So as an example, when we look at our panel, we currently get about 60 or so variants per person. That's not too difficult for us to investigate and see whether they have good coverage, whether they're an artifact, whether it's just a common polymorphism or whether it's a mutation. But if you want to do the whole exome, so doing all the genes, you pick up over 35,000 variants per person. So you get a lot more things that you have to investigate. And it's quite difficult. So what we've done is also set up a bioinformatic pipeline that allows us to prioritize which genes that we're investigating and allows us to filter them and to identify which could be potentially playing a role. So we prioritize this based on how common it is. If it's a causative mutation in a family, it's unlikely to be a common polymorphism. So how common it is important for us, and that's one of our ways of calling a variant. The second thing is we look at when we do curation and whether we potentially define a potential variant, we have to undertake analysis according to the American College of Medical Genetics guidelines. We look at the literature to see whether a particular variant's been examined previously. We look at all the known databases to see if there's been anything that mentions this particular novel or whether it might be a known variant. We also use in silico tools to determine whether the variant we've identified is potentially mutagenic or not. So is it likely to be pathogenic and affect function? And then we curate those based on those guidelines and provide a diagnostic result that goes out. We're not the only lab that has done this and does this. So as I've mentioned, there are guidelines for how this is done. I'm just giving you a bit of an overview of the process that's involved in this. So that's the current method that we use for investigating diagnostically. But what happens if you actually have looked at all the potential known genes and you haven't been able to pick anything from a panel or from known genes that could be potentially identified through whole exome sequencing? Well, what we've done is also, and others have been doing, is undertake whole exome sequencing in those unsolved cases. Recently, we looked at 170 clinically diagnosed cases that were not positive, so they had no mutations in the known genes. We also looked at family members of these, and we did whole exome sequencing on over 200 cases or individuals. And from that, we were able to identify a diverse range of potentially pathogenic rare variants in these particular families. Even though we didn't pick up what was considered to be a recurrent mutation in the PRRT2 gene, we did pick up some other pathogenic mutations in this gene. We also picked up some mutations in some other novel genes for hemiplegic migraine, including all the ones here. And basically, we added about another 10% in terms of our diagnostic hit rate into these. Most of the mutations detected in the genes are private mutations, meaning they're a particular mutation that occurs in a specific family. And it's not necessarily common to a range of different probands for FHM. One particular area we've been focusing on recently in terms of the analysis of this data is to be looking at calcium channel genes, and one in particular has identified rare variants in the CACNA1I gene. And we've shown that burden testing within this showed a statistically significant increase in variants within this gene in FHM versus controls. And we looked at controls from NOMAD and also the UK Biobank with very significant control populations there. Since dysfunction in T-type calcium channels has been implicated in neurological conditions, we decided to then investigate these. So we did some functional studies investigating patch clamp electrophysiology in cells there. And we found that numerous functional alterations, including reduced current density, voltage changes, and also current kinetics as well. So we've recently, in fact, quite recently published this in Frontiers in Molecular Neuroscience, but at this stage, it looks like there's variations in this gene that could be playing a role in FHM, whether it's a causative role or whether it's a modifying role. We're not absolutely sure yet, but there's certainly some results there that seem to imply that it's not just the CACNA1A gene, but there may be other calcium channel genes that are playing a role in FHM. Our continuing studies here and other continuing studies that others are doing around the planet is to focus on diagnostic testing and extend this as we identify our variants and genes from our research and put that to full diagnostic use. We're continuing to analyse our whole exome sequencing data to try and identify genes that play a role in FHM. We're also undertaking similar studies in episodic ataxia, CADASIL, and some epilepsy-related disorders as well. We're currently expanding functional analysis, but also familial segregation, so testing these in families. And I think the other thing that we really need to be thinking about in terms of genetics of migraine is that not everything will necessarily be single point mutations. We need to be looking at other methods of detecting other particular types of mutations, such as those that are not just in exons, but maybe within promoters, maybe in enhancers, non-coding areas, or even things like copy number variants, structural variants. And so we're currently looking at those areas as well. Another, just towards the end here, I want to mention a couple more things, but probably another area if we want to think about the genetics of migraine is to think about post-traumatic migraine genetics as well. And in particular, this has been sparked by information that's made it clear that individuals with a specific CACNA1A mutation have really high sensitivity to bad outcomes from head impacts. So even minor head trauma can lead to seizures, cerebral edema, coma, and sometimes even death. And we've published on this a number of years ago, but many others have as well. So this is rare, but individuals with these mutations, as I've mentioned, can have some quite significant outcomes from minor head trauma. So this led us to start looking at other ion channel genes in relation to things like susceptibility to concussion. So of note, we started looking at other FHM-related genes to see if there was a potential role for this. And we found that there were notable characteristics in patients who had mutations within the ATP1A2 gene, that is the FHM2 gene. So when we looked at those with of 172 of our patients, we found that of those, 68% of those were due to mutations with ATP1A2. So, and I think more importantly, there was an overrepresentation of patients in the ATP positive group compared to the negative group who had noted mild TBI or concussion in their clinical notes and clinical background. And this was associated with specific mutations within that gene So as I've mentioned, looking at those particular mutations within ATP1A2, they're associated with mild TBI-induced concussion in 33% of ATP1A2 positive patients. So the results of that, but also the results from the CACNA gene, which was identified quite some time ago, has led us to start thinking more about whether ion channel genes could be potentially playing a role in post-traumatic migraine as well. And so we've currently been undertaking whole exome sequencing to see if there are rare variants in other ion channel genes that could also be playing a role. And those studies are continuing. So towards the last minute, I would just like to mention probably two other areas that are important to, I believe, are important to the genetics of migraine. One of these is epigenetics, as I mentioned a little bit earlier. So epigenetics involves heritable changes in gene expression, and these occur in other mechanisms other than actual DNA sequence changes. So it could be modifications within the histone proteins that are important for packing the DNA shape into chromatin. It could be variations within the histone genes, could be microRNA, or it could also be DNA methylation, which has been known to play a role in gene silencing. So epigenetic factors are heritable. They're changes that can affect gene function, and they do not involve changes in the DNA. So probably the most commonly investigated one is DNA methylation, and we know that it's modifiable and it can actually change across the lifespan, and it's also is inherited. So it's also been associated with a number of disease traits. There's been a few studies looking at methylation patterns and epigenetics in migraine, but not a great deal. So some of those have involved quite small numbers, and there's one here by Gehring et al., a couple of others as well. And they've implicated potentially some genes, but no significant results really outstanding at this stage, although there's a few implicated potential disease genes there that may be playing a role. So even though the migraine epigenetic studies to date have been quite limited, this is an area that's certainly worthy of investigating more in relation to things like migraine development, potentially the subtypes, and even into treatment response. The last area I'll mention that I think we should be exploring in relation to migraine is pharmacogenomics. So this is the study of differential response to medications due to different genetic variation backgrounds. The goal of pharmacogenetics is to develop individualized treatment strategies that are based on genetic makeup. Now, since not all patients respond well to different migraine treatments, perhaps pharmacogenomic studies can help us define the most appropriate treatments. There's been a number of papers that have outlined the potential importance of this, of migraine pharmacogenomics, but there's been very few studies so far. So the treatment of monogenic forms of migraine, as I've mentioned, and related disorders such as FHM and PRRT2 already utilize genetic mutation results to aid in treatment choice. So this could be further developed and applied to common types of migraine to help understand things like variability in response to treatments, and even to develop accurate predictive panels for acute and preventative treatment response. And if we could do that, that would really help in individualizing migraine treatment based on genetic information. So in conclusion, sequencing has become a cost-effective approach, not just for migraine research, but also for diagnostics, to identify the genes involved in the various types of migraine. Many genes and genetic variants have been implicated, particularly in the monogenic forms of migraine, and there's ongoing studies to improve diagnostic success rates. For the more common types of migraine, also many genes and genetic variants have been identified, but this doesn't yet account for all the hereditary factors involved in the more common types of migraine. So there is a need for future research to do that. Potentially investigating rare variants, structural variations such as copy number variants. Also investigating epigenetic factors may also help account for this missing heritability in migraine. And finally, genetics also holds the promise of identifying predictive pharmacogenomic profiles, which could aid in developing more individualized treatment choices. for migraine management. I want to acknowledge all the people that work with me in my lab, both the genotyping, bioinformatics and diagnostic staff, also our clinical collaborators and contacts, and also the support that we've had from a variety of organisations for migraine research. And I just want to thank you for your time.
Thank you very much. This was very informative and very in-depth explanation of all the work. And thank you for all the great work that you're doing to help us understand migraine a little bit better. My question for you, I have a couple of questions from attendees. And the first one is, how close are we to finding a test kit or a diagnostic tool that will diagnose migraine in general? How close are we to finding one?
Okay, so right now, as I've mentioned, we do have a diagnostic test kit, and there are a number of testing labs in different parts of around the world that do diagnostic testing for the rarer, severe monogenic forms of migraine. And as I tried to mention today, I think it's important for diagnosing those disorders so that you know what you're looking at, and it's also important for treatment choice, also to rule out other disorders. However, I think we're still a fair way away from having a simple diagnostic test, gene-based one for migraine. As I've mentioned, there is still some missing heritability there. We've been able to develop polygenic risk scores, which should aid in this, but I don't think we're quite there enough. We are getting there though, and collaboration is the absolute key here in terms of getting the numbers of cases and controls and sufficient power. But I think the other thing is to realize it's not going to all be straight genetic variants. We really do need to bring in structural variants, which means we're going to need to do some whole genome sequencing with those sorts of variants to bring that in. And I think we also need to be looking at things like epigenetic markers. So I'd love to say it's next year, but I still think it's a few years away for the common types of migraine in terms of a simple genetic test.
And in terms of, you said that cost and time are big barriers at this point of time to making this available to everybody. Also, if they want to send just out of curiosity, I guess, what are we talking about? How much does it cost for someone in Australia, obviously, or in your lab to get tested?
I must say, when I first started doing diagnostics, well, we started looking, as an example, at the CACNA1A gene. To use a Sanger sequencing approach for the gene there, just one gene would have cost $4,000.
Wow.
And it was very time consuming as well. So now we can, as I've mentioned, we can do 15 genes at once. It's a targeted panel, and it costs around about 500 Australian dollars in our lab. And that means, right from getting the sample to doing all the analysis and getting the result back. I think it's coming down all the time as well. So a while ago, we were charging more for that, but the technology is getting a lot better. Whole genome sequencing can now be done, and there's this for under $1,000. The problem is that there's so much data that comes out of it that you couldn't analyze all of that for that same amount of money. So you might be able to sequence it, but the analysis time is enormous. So I think at the moment, the most sensible thing for migraine is to look at targeted panels. If you're looking at some of the severe types of migraine, I think it's much easier to focus on those particular genes, and you're more likely to get a diagnostic outcome that makes sense. However, whole exome sequencing is also available and at a reasonable price as well. It means that you're covering many other genes, particularly if it's not within the more common targeted panel. So it depends how good the clinical diagnosis is, and the family information, I think, as to which is the best method, targeted or whole exome. But I think it's within reach, and it's going to keep coming down in terms of costs.
I have a very interesting question here. Do you believe that genetics will help us identify the etiology of migraine, a more universal etiology, instead of, we know that there are so many etiologies now, or pathways that are linked to migraine, but we don't have a central cause why someone would.
I think that's exactly what the promise of genetics is all about. So it's the more we learn about the genes that underlie it, we know which pathways are involved, and we can then look at new targets for treatment. And it's already given us some clues about targets for treatment now. But I think the more we learn, we're probably missing, because we haven't got all of those genes here, we're probably missing some of the pathways that we could potentially use as targets to develop new treatments and understand etiology. But we could also be looking at individual variation as well. So at the moment, when people are given migraine treatments, it's a bit hit and miss in terms of which one people should be given, whether it's going to work for individuals, whether there's going to be adverse effects. So as I mentioned towards the end here, that's where pharmacogenomics is going to come in. That's within reach right now, to be doing profiles on individuals and to then see if we can identify whether those profiles will be able to target the appropriate treatment for individuals. So etiology is one side and can potentially give you new treatments, but I don't think it's going to be one treatment for everyone. I think using pharmacogenomics, we'll be able to define more individualized treatment for an individual.
Well, now that you mention pharmacogenetics, I know in Canada we do have the possibility to do pharmacogenetic testing or genomic testing before we initiate a treatment. The thing is, they don't have all the new ones there for us to actually look at. And these are the most expensive and the ones that possibly we would need to test for, just to avoid that someone would be using thousands of dollars for no reason. But also we find that it does predict more tolerability, but it's not really as close. I don't know if you can also shed some light on this. I don't know if I would send a patient to do this before I try.
I think, yeah, Hiba, I think at the moment, most of the pharmacogenetic testing that's routinely done or done when people are considering tend to be based on how drugs are going to be metabolized and whether that's appropriate. Occasionally, there are some that relate to specific disorders. In terms of migraine, I don't think that's so important at the moment. What we should be thinking about, I think, for pharmacogenetics and migraine is really defining what is the profile of an individual that would fit well in terms of treatment response. So as an example, CGRP treatments work very well for some people. Botox works well for some people. Triptans works well for some people. We should be looking at the profile that relates to which of those, so that it's not a hit and miss as to which you should be having. So it's not so much the metabolizing genes that we're looking for adverse effects. We're looking for what's the appropriate profile to decide predictively which will be the right treatment. And I think as we define more treatments, as we get more etiology and target markers, we'll have more treatments, but this will be the thing that will go hand in hand with defining that.
Thank you very much. I know it's very late. Is it after 3 now where you are, 3 a.m.?
Yeah, it's just after 3 a.m. and I'm not sure I'm making a lot of sense to everybody at the moment.
No, you've made more than great sense to all of us. Thank you very much. There's still a lot of questions, but I'm going to say thank you very much. Thank you to IVPN and the Association of Migraine Disorders, and most importantly, our sponsor. So this series has been very generously sponsored by Pfizer, our sponsor for this event. So thank you very much. If there's anybody from Pfizer tonight with us, thank you very much. And again, Professor Griffiths, it's been an honor having you, and we look forward to reading more of your amazing research on this topic. And it's always a pleasure to hear all these advances, and the future is bright, I think, because of efforts in this field. So thank you very, very much for your time, and
You're
have
most
a very
welcome,
good night.
Hiba. And I just want to say thank you very much to Pfizer, and thank you very much to all the audience as well. It's a funny way to do this when you can't see all of your audience, but I can see that there's a significant number of people out there. So I hope this was of benefit. And also thank you very much for inviting me to be part of this. I will go to sleep now, though.
And I will tell you, you're more popular than a soccer game or a football game. It's a big game now, and everybody is here who's really committed, and they've stayed. So thank you very much. Have a very, very good day.
Thank you. I hope the rest of the event goes well. So I'm going to disappear now, but bye, everyone. Thank you.
Okay, thank you very much. I can see there's still some questions that we haven't answered. We will do our best to answer at the end. Of course, our next speaker is from our region. I'm very happy to introduce Dr. Tawfiq Al-Saadi. I've had the pleasure to working with Dr. Al-Saadi years and years ago, professional knowledge, everything that you would want in a speaker, and I'm very happy to have him join us today. So he's going to talk to us today about new comprehensive approach for migraine management. Needless to say, he is the chief medical officer and chair of the neurology department at the American Center of Psychiatry and Neurology. He was trained in epilepsy and clinical neurophysiology at the University of California, San Francisco, and was a research fellow in epilepsy there. He is board certified by the American Board of Psychiatry and Neurology and the American Board of Clinical Neurophysiology and by the American Board of Neurology in clinical epilepsy. Before joining ACPN, Dr. Al-Saadi was the chief of neurology department at Sheikh Khalifa Medical City for 11 years. He previously held the position as an associate professor of neurology and the director of the adult epilepsy program and the clinical research program at the University of California, Davis. His research has focused on conducting drug study trials for new anti-epileptic drugs for patients with epilepsy or people living with epilepsy, quality of life in patients with epileptic and non-epileptic disorders, migraine headaches, multiple sclerosis, neuropsychiatric comorbidities, and many, many more. Dr. Al-Saadi has been honored with several prestigious awards for his work in the field of epilepsy. He has served on the advisory board of the Northern California Epilepsy Foundation and on the membership committee of the American Epilepsy Society. He has previously served as the president of the Western Clinical Neurophysiology Society in the US and as the vice president of CIMA or KIMA, which is the Commission of the Eastern Mediterranean Affairs. He is currently the president of the Emirati League Against Epilepsy. He also serves at the editorial board for BMC Journal of Neurology and Journal of Neurosciences. He has authored and co-authored more than 65 papers and book chapters and has served as peer reviewer for more than 15 scientific journals. Welcome. I know I can spend more time introducing you, Dr. Al-Saadi, and it's such a pleasure to see you here with us tonight again, while there's a big game playing. Thank you for giving us the time and giving us your knowledge today.
Good evening, everyone. Thank you very much, Hiba, for the kind introduction. I know I'm giving this lecture at a time where there's an important game playing right now with Brazil versus Switzerland. So I'll do my very best to be as quick as possible so people can probably try to get to at least part of the game. So what I will be covering in the coming 35 minutes or so, I'll try to cover some of the new areas in the management of migraine, specifically with the new molecules that have become available to us. So I'll start off with my disclosure. And you can see my slides very well?
Yes, they're working perfectly okay. Thank you very much. Sorry, I had my mic on. I hope I wasn't.
Well, we know that headache is the most common disorder that we see in our day-to-day practice as neurologists, as well as other healthcare providers, with migraine being one of the most common headache disorder that we see in our practice. Globally, we know that at least 1 billion people worldwide had migraines. In the States, about 40 million people had migraines. I would say, unfortunately, it's a disease that's more common in women than men, where it's almost 3 times more common in women as compared to men, with at least 20% of women in the States are impacted as a result of migraines. Well, wherever you practice, in the States, Canada, this part of the world, migraine is a highly prevalent disorder with estimates in the range of 11 to 15%, again, depending on which study you are looking at. Now, how does it compare to other chronic disease states? as we can see from this slide, that migraine is far more common as compared to other chronic disease states, including asthma, diabetes, and osteoarthritis. Now, how does it compare to other chronic neurological disorders? Also, we can see that migraine is far more prevalent as compared to Alzheimer's disease, stroke, epilepsy, or even multiple sclerosis. Well, more importantly, if you look at the 2016 global burden of the disease, you can clearly appreciate that migraine is the second leading cause of years lived with disability. But if you look at those in the productive years, meaning from the age of 15 to 49 years of age, we can see that migraine take the lead in terms of years lived with disability. It certainly, we know that migraine impacts your life at multiple domains: your physical function, everyday activities, social life, relationship, as well as emotional responses. According to the WHO, it stated that a day with migraine is equal to a day with patients with quadriparesis, psychosis, and dementia. Well, that's obviously not surprising, knowing that at least 50% of patients with migraine have severe disabling attacks. The problem we face with patients with migraine is polypharmacy. According to this cross-sectional study that included 300 patients, 80% of them with headaches, 80% of them had migraines. Look at the average number of headache medications taken by a patient is around 4, with a daily number of medications taken by at least 40% of patients more than 5. So polypharmacy is an important issue in patients with headaches in general. Now, in terms of management, there are 3 concepts that we need to bear in mind. That is very important for a patient with migraine to look at these patients from a holistic point of view, meaning that you cannot look at patients only with headache. Headache is only one component of the story, but oftentimes these patients have other comorbid conditions that you have to address in order to have the best outcome of treatment. So we always look at non-pharmacological approaches, acute therapy, as well as preventive therapy if patients are candidate to that therapy. I'm not going to spend too much time on non-pharmacological options, but I can tell you, at least based on some evidence-based data, there are several strategies that have been shown to be quite effective in patients with migraine, including biofeedback, cognitive behavioral therapy, acupuncture. Those are some of the measures that have proven some efficacy based on good randomized controlled trials. Now, if you look at the acute therapy for patients with acute migraine attacks, well, according to the American Headache Society, that there are certain criteria that had been adopted by the American Headache Society that every patient with migraine should be offered that trial of acute treatment that would offer rapid and consistent freedom from pain and associated symptoms such as nausea, vomiting, and photophobia, restore ability to function, minimal need for dosing or rescue medication, reduce obviously in healthcare resources, and very importantly, minimal or no adverse events. Now, if you look at the current limitations of the currently available medication, at least until a few years ago, obviously we have issues related to the tolerability of the side effects profile of some of these medications, specifically triptans, where almost half of these patients may not be able to tolerate triptans. At least a third of them would need to use a rescue medication. Pretty much all the previously available acute therapies are agonists, so there's always risk of medication overuse headaches in our cohort of patients, especially those who are heavily using these medications. And some of our patients with migraine have some cardiovascular risk factors, and that would obviously limit the options of some of these therapies. And that's what obviously had called to find newer approaches to managing patients with migraines, looking at distinct mechanisms of action in order to avoid some of the problems we had with the previous available medications. So specifically those with the triptan responders. We know the issue with the triptans being vasoconstrictor, so we need to find a drug that does not have that problem, longer half-life, and very important, as I mentioned earlier, where medication overuse headache is quite common, you need to find drugs that do not have that agonist approach meaning they can be work on different receptors, being antagonists, so they don't have the problem with medication overuse that we typically have seen with the triptans and other medications for acute therapies. So if you look at now at the map of the currently available options for patients with acute migraine attacks, we can call it into two columns, those with migraine specific and those with migraine non-specific therapies. For the migraine specific therapies, the older medications, we have the triptans, ergots, and lasmitamide, and the newer drug now had become available to us were the CGRP small molecules, with specifically GPANs. Those had become available to us. On the other hand, we have what we call non-migraine specific medication, meaning they work for patients with migraine, but also they work for other headache types, such as the NSAID and other medications. They work for migraine as well as for other headache types. So these are non-migraine specific medications. And now what about preventive therapies? Obviously, patients with frequent attacks, they need to be offered migraine preventive therapy in order to reduce the severity, the frequency of the attack, and minimize the risk of going into the medication overuse headaches. And this is some of the criteria we look at in order to offer these patients migraine preventive therapy. Obviously, has to do with the attack frequency as well as other parameters, including the severity and the restriction as a result of these attacks, meaning the disability score. So it's not only the frequency, but how disabling these attacks are. These some of the things that we look at before we offer these patients migraine preventive therapy. The problem with the current map of treatment with a patient with migraine that, yes, 40% of patients with migraine can be a candidate for migraine preventive therapy. Only 1 in 10 of them are offered such a therapy. And the problem with the previously available preventive therapies that at least half of our patients may not continue on these drugs. And if you continue to follow up with these patients up to 6 months of treatment or even longer, probably 1 in 10 would remain on that treatment. And that is regardless of whatever preventive therapy you choose, being anticonvulsants, antidepressant, or beta blocker, at least 1 in 10 would continue on that therapy if you follow these patients up to 1 year of follow-up. So we have several limitations of the previously available preventive therapy. They certainly take quite some time before they can be, we can see some efficacy, at least several months before we see some response. We have issues related to their poor tolerability, their side effects profile, specifically issues related to weight gain, mood, especially can be quite a problem in patients with migraine, knowing that at least 3 times more common in women as compared to men, as well as higher discontinuation rate, as I alluded to earlier, that 1 in 10 would continue on that therapy. And finally, obviously, patient's preference would take into account in terms of which medication we choose. But we need to keep in mind there's so much variability in the attack frequency in patients with migraine, with almost 80% of our patients with migraine have low frequency migraine attacks, and about 15% have high frequency episodic migraine, and about 5% or so, they have what we call chronic migraine, which 15 days or more with headaches. So you have to keep in mind there is so much variability in the attack frequency within individuals and across people with migraines. So obviously with patients with migraines, polypharmacy is a major issue. Patient preference is something to look at before you offer these patients some form of treatment. And it's preferable probably to choose a drug that can tackle both of these problems, meaning acute therapy as well as preventive therapy. And the only drug that we have so far that has been recently approved by the FDA for the possibility of tackling both acute as well as preventive therapy is with rimegepant, that has been approved by the FDA for both of these indications. We all have heard about the story of CGRPs and its role in patients with migraine. This is what happened during the resting state. The CGRP levels is down, and then during the migraine attack, the CGRP level goes up. If you infuse CGRP into patient with migraine, that can trigger a migraine attack. And the selective binding of the CGRP receptor antagonist treats migraine with the advantage, meaning that it does not have the vasoconstrictive effect that we have seen before with the previously available triptans. Plus, you have to keep in mind that these drugs, they work as an antagonist. There is no risk of medication overuse headache that we have previously seen with the previously available triptans. The approval of this medication in both of these indications had come after two main pivotal trials, the 303 trial in patients with acute migraine therapy, as well as the 305 trial for patients with chronic migraine. And then these patients had went into the longer term extension studies. For the 303 trial, they looked at patients with moderate to severe acute migraine attack who were randomized to either the medication, Nurtec or Rimishpant versus placebo. And they look at main primary endpoints, which is the freedom from pain at 2 hours dose, as well as freedom from the most bothersome symptom, which are the recent adopted guidelines by the American Headache Society, the International Headache Society, as well as the FDA. And they have also some secondary endpoint results, which we'll get to later on. And if you look at the demographics of their cohorts, obviously, most of these patients are women. About 70% were migraine without aura and about 30% migraine with aura. Mean age is around 20. If you look at the historically, the most bothersome symptoms, which is very important to keep in mind, 60% had photophobia, about 20% had nausea, and they have other most bothersome symptoms in their cohorts. And if you look at the primary endpoint, which is again freedom from the most bothersome symptoms, as well as the freedom from pain, if you look at this study, clearly statistically more significant patients had achieved that primary end goal in the treatment arm with the Rimishpant or Nurtec as compared to the placebo in both of these primary and co-primary endpoint. If you look at this slide on the left-hand side, you can see that the separation of curves start at half an hour, but it became quite statistically significant, 1.5 hours, where more patients who were randomized to the drug had more pain freedom as compared to the patients who were randomized to the placebo. Now, if you look at what we call the patient's pain relief, which was the primary endpoint that was previously used by the previously available medications, 2/3 of these patients had achieved that secondary endpoint at 2 hours as compared to the placebo arm. And if you look at the ability to function normally with medications compared to the placebo, almost 40% had achieved that goal after 2 hours as compared to patients who were randomized to the placebo arm. More importantly, how likely the patient will have the sustainability of the pain freedom. You can see, obviously, more patients on the drug had sustained pain freedom up to 48 hours as compared to the placebo arm, sustained ability to function normally also up to 48 hours, statistically more significant patients on the treatment arm as compared to the placebo arm, and the use of rescue medication was way much less in patients who were randomized to the treatment arm as compared to the placebo arm. More important, if you look at the side effects profile, the drug seems to be quite tolerated with very minimal side effects profile, namely nausea, urinary tract infection, but quite minimal, or less than 2% of the patients who were treated on the medication as compared to the placebo arm. So if you look at the summary of the trial on the acute migraine therapy, obviously significant greater proportion of patients on randomized to the drug had met the primary as well as the secondary endpoint in the pivotal trials. The drug was well tolerated and with a good safety profile, and most common adverse event was reported in less than 2% of patients were basically nausea, urinary tract infection, and dizziness, but more importantly, there were no serious adverse events that were reported in that trial. Now, the second trial that had led to the approval of this medication in preventive therapy was the 305 trial, where adult patients above the age of 18 with more than 4 but less than 18 migraine attacks per month were randomized to either the drug on every other day basis versus placebo, with the primary endpoint with the change in monthly migraine days from baseline from the week 9 to 12 as compared to the placebo, where there's also some secondary endpoints we'll get to later. And then the patients went to the open label phase of the trial, which had lasted up to 52 weeks. Again, looking at the demographics, similar to what we've seen in the previous trial, mostly female, migraine with aura is about 40%. Median duration of untreated attacks was around 24. median age at disease onset was 18. History of chronic migraine was 20% of these patients had a chronic migraine. Now, if you look at the primary endpoint, which is the main change from baseline in monthly migraine days, were obviously statistically more significant in patients randomized to the medication, rimegepant, on every other day basis as compared to the placebo arm. If you look at the other secondary endpoint, such as the responder rates, especially in the first 3 months, obviously again more in favor of the medication as compared to the treatment, the placebo arm. And if you look at the slide, again, change from baseline in monthly migraine days, you can see the separation of curve as early as 1 month and remain up to 3 months of continuous treatment. If you look at the side effects profile, similar to what we've seen with the 303 trial, the drug was very well tolerated, minimal side effects, nausea and nasopharyngitis being reported was less than 2% of patients, but there's no serious adverse event was reported, with no serious events in terms of abnormal liver function abnormalities in the trial as a whole. So in summary of this particular trial, the rimegepant on every other day was significantly different from placebo on the primary endpoint of change in the mean number of migraine days per month during the weeks 9 to 12 relative to the initial 4 weeks observational period. It also had a benefit in terms of proportion of patients becoming responders, meaning more than 50% reduction in mean number of moderate or severe migraine attacks in the last 4 weeks of the 3 months double-blinded treatment arm, and again, no serious adverse event had been reported. These patients had been allowed to continue on extension phase of the trial, where over 112,000 doses of the drug were administered across 1,800 patients over a year, and again, similar safety profile that we have seen, and making us more reassured about the safety profile of the drug, with no clinically relevant lab abnormalities were observed. This is how the drug was administered, either as a cure therapy versus as a preventive therapy on every other day basis. Obviously, you have to pay attention to the drug-drug interaction if patients are taking cytochrome P3A4 inhibitors, and the contraindication if patients are hypersensitive to this medication. Obviously, precautions in patients who are pregnant or breastfeeding, as well as in hepatic and severely renal impaired patients. Let me walk you through some of the cases that I've seen in my practice, and I'll give you some idea of how we deal with some of the cases that we see in our practice in neurology. So we have a 30-year-old woman with history of frequent attacks of headaches for the past 10 years. She had no aura before the attacks. The attacks were moderate to severe in severity, quite debilitating. The attack can occur mostly during the daytime, with peak almost in 33 to 45 minutes. She averaged about 2 to 3 attempts of attack in a week, with each attack lasting up to 2 days. Oftentimes, these attacks are associated with strong nausea, photophobia. She has also comorbid depression. At the time I saw her in the clinic, she had been tried on a number of medications, but she was only at that time tried on NSAID. She tried on other medications. They had inconsistent response, meaning some of the attacks had responded to triptans, others were not. At the time I saw her, she was on flunarizine 10 milligram, previous trial on other tricyclic antidepressants, as well as topiramate, were unsuccessful. Obviously, due to the severity and frequency of the attack and due to her work, she had to call in sick, unable to attend to her meetings, her work assignment, due to the severity of these attacks. Obviously, we're dealing with a woman with highly episodic migraine headache, and that will give us some options how to manage this patient. Should we continue the drug that she's currently using, which is flunarizine? Should we continue and consider other preventive therapy? But regardless of what decision we make, continue or discontinue flunarizine, what are the options if you want to consider for preventive therapy? At that time, we had the privilege of having an access to the monoclonal antibody. So we started her on one of the monoclonal antibody, and we plan to discontinue flunarizine, provided that she had good response to this monoclonal antibody over 3 months of therapy. So she comes back after 1 month. She was quite happy, had 1 month attack, quite happy with that. But on the following months, she reported several attacks that were not responding to several medications, and she had to go to the emergency room receiving IV analgesics. And that will give us some options how to manage these attacks. Well, obviously, you can try some other triptans. So she tried several of them. Lasmitamine is an option. Or can we try dihydroergotamine, or should we try another option? Obviously, before we address some of these points, we have to remember some of the points, such as the consistent results of the triptans, the timing of the attack, remembering that most of our attack occurring during daytime, and we considering some of the options that we have with the newer medications. If you look at the story of the triptans, obviously we have a problem with the consistent response with triptan. This is the data from Richard Lipton. If you can look at the likelihood of patient becoming respondent in 1 out of 3 attacks, only the best we can see with rizatriptan with almost 70% had consistent response. Whereas if you look at the pain freedom in 1 out of 3, only again 50 or 60% had that endpoint. If you look at the response in 2 out of 3, again, the best was seen with rizatriptan, with almost 60% are responders. If you look at the pain freedom, 50%. And if you look at 3 out of 3 responders, less than 50% in the case of rizatriptan, and less than 20% if you look at pain freedom. So the consistent response is quite of a problem in general if you are to consider triptans. And that is also the case with the newer medication that had become available to us with lasmitamine. And that will take us again the option of other medications, especially bearing in mind that most of our attacks occurring during daytime. So if you are to consider lasmitamine, for instance, that could be a problem because there are certain warning about using this medication because of restricted driving after 8 hours after taking this medication. So you have to bear this in mind if you are consider lasmitamine. So what we did with these patients, obviously we had again the privilege of having an access to the small molecular CGRP antagonist, the GPANs and the Migraine. So we had offered that patient this medication, and this is some of the results that we have previously shared with you. And she tried on this medication. We saw her follow-ups after she had to try this medication on several occasions with very good response, with no reported side effects had been at least shared with us from our patient. Now, let me walk you to another problem we see with our patient with migraine. This is another woman patient with 32 years of age with meso-related migraines. She had migraines for the past 15 years. Attacks were moderate, severe in severity. They tend to occur almost 2 to 3 days before her cycle or 1 to 2 days after the cycle, each attack lasting 1 day or so. Strong nausea, occasional vomiting. She has no other past medical history. At the same time I saw in the clinic, she tried on NSAID. She previously tried on sumatriptan, zolmitriptan. They were quite ineffective. She also tried other medications for meso-related migraine prophylaxis, including neurotriptan, fluvotriptan. She had also tried one of the monoclonal antibodies for migraine prophylaxis for 6 months, but also that was ineffective. So what should we do? Should we try another monoclonal antibody? She tried one that failed. Should we try another one? Should we try a long-acting non-steroidal agent for meso-related migraines, or should we try something else? So at that time, again, we had the privilege of having the availability of the rimishipran GPANs available to us. So we offered her that medication 4 days before the cycle, 3 days after the cycle on every other day basis, with continuous follow-up with her 1 month and then several months after the initiation of therapy. She reported extremely very good response with 1 or 2 occasional headaches during the menstrual cycle, very mild, not needing to take any painkiller for these attacks. And finally, let me walk you through this last case, which is a 35-year-old woman with history of headaches over 15 years, again, averaging 3 attacks per week, each attack lasting 1 to 2 days, has comorbid depression, never tried on any preventive therapy. Though she had headaches 15 years, which by the way is quite of a problem, not only this part of the world, even the state, as I mentioned earlier, 40% can be candidate for migraine prophylactic therapy, but only 10% or so are offered that therapy. When we saw her, she stated that she's needle phobic, so that could again limit some of the options for migraine prophylaxis. Should we go for that option? And again, due to the severity and frequency of the attack, she had to call in sick, unable to attend to her work duties due to the severity and the frequency of these attacks. Obviously, again, we're dealing with a woman with a highly episodic migraine headaches, and then that will take us to the option, what sort of preventive therapy we can go on this patient. Well, obviously, we have so many options, including some of the oral available medications, as well as the injectables, but you have to bear in mind that she's needle phobic, as well as the currently approved medication that we have previously discussed, rimishipran for migraine prophylaxis, that had been shown, I've shown you the data on every other day basis. And after I laid out all the options to her, she elected, after lengthy discussion, to go on rimishipran on every other day basis. And we saw her on regular follow-up visit from 1 month after the initiating on treatment, and then up to several months after starting on treatment. And she continued to do extremely well, reporting very occasional attacks, that most of them they would not need to take any painkiller. And that's actually a measurement of how successful your treatment is when the attack frequency becoming less in terms of severity, frequency, as well as you need to take any rescue medication would become exceedingly less. That obviously indicate that the drug, whatever drug you choose for migraine prophylaxis, is extremely working that well. So with that in mind, I will leave you with final thought. Obviously, the rimishipran has been approved by the FDA for acute migraine therapy as well as for migraine prophylactic therapy. It has a unique, distinct mechanism of action targeting CGRP. It's offered sustainable efficacy in both patients during migraine attack as well as for long-term prevention therapy. The drug is well tolerated with very minimal reported side effects and very minimal discontinuation rate that have been reported in the literature so far. With that, I would end, and thank you very much for your attention. I'll be very happy to take any questions after this. Thank you very much.
Thank you very much. This has been very interesting and very informative. We really thank you for sharing your slides. A quick, I have a few questions. I think, yeah. Are we able to stop sharing from your side, Doctor?
Let me try this. Yes, I can.
Perfect. And we have Maya joining us again. So a few questions are coming around. Currently, there are no guidelines about how you can integrate, or no unified guidelines. There's a lot of suggestions and possible proposals how we can integrate the new options that we have now. So the question is, do you start from the beginning? Are they first line for you? Or do you still go ahead and start with other options before you go for the new options, whether CGRP, monoclonal antibodies, or GPANs?
Well, there were some guidelines before. There was the European guidelines, the American guidelines, which at least until very recently, that suggested that you need to try at least 2 what we call standard of care therapies before you can offer these patients a treatment with the CGRP antagonist. But that was the case until very recently, when we have the European Federation Council guidelines that has suggested CGRP antagonists can be offered as first-line treatment in patients with migraine. So that came up just very recently, that patient with migraine can be offered that treatment as first line before consideration of failure of other medications. So this is very recent. This, again, according to the European Federation Council, that sort of treatment can be offered. Obviously, in my practice, obviously we're practicing in UAE, and we had the privilege to have access to these medications. As a matter of fact, we were the second country after the state to start using this medication. So we have very good data on these medications, and we have currently 3 approved CGRP antagonists that are available in injectable forms, and we have now 2 of the approved medication GPANs. One of them, rimishipran, which is approved for both acute and preventive, and another drug, rimishipran, which is approved for acute migraine therapy. All of them are available to us in UAE, and we have the privilege to use these medications. And obviously, remembering that some of our patients are Emirati locals, and we have no problem in terms of accessibility, in terms of insurance approval in these patients. So that gave us obviously the privilege to start using this medication as first-line therapy. And I believe, and as a matter of fact, even before the European guidelines became available to us, we in UAE, knowing that we finally got to have an access to migraine-specific therapy, there was no point, obviously, to consider these what we call standard of care therapies, which we don't really know how they work in terms of mechanism of action. We have a problem with their side effects, tolerability issue. With the availability of the newer drug, doesn't make any sense to move on with the previously available medication, knowing that we have better tolerated and far more effective medications with the CGRP antagonists, whatever which drug you choose, after these drugs have become available to us in UAE.
And I have a question in the session here. Someone asking, when it's not available, what do you do? Like it is available, luckily, in some countries. In Canada, we still don't have the GPANs. We're expecting them next year. So when GPANs are not available, what would be, how would you deal with this? They're asking, what other medications do you use? Of course, we have to separate by acute and by preventive, I guess. So let's start with acute. Do you have a process where you decide which patient gets which treatment? I know that there are certain algorithms, but how do you do this practically?
There's, as you said very correctly, there's an algorithm. So the two choices of treatment have to be individualized. We have to look at the patient's profile, patient's preference, the specificity of the attack itself. And for instance, I gave you an example before that one of the patients that I have stated earlier, the attack occurred during the daytime, so that would limit some of the options of treatment. Time to peak, this is very critical as well. Time to peak of the attack, and that particular patient said the time to peak, it's from 3 minutes up to 45 minutes. So time to peak is quite fast. So you need to choose a drug that acts very fast in order to get the best result. Whereas in other patients, the time to peak can be several hours. So you can use a drug that it has longer half-life. It tends to peak later in time, like naratriptan or forever. So the choices of drug is quite dependent on several factors, comorbidity factors, as well as the characteristic of the headache itself. So for instance, patients with cardiovascular risk factors, for instance, I would not choose the triptans. I would probably be very careful with using triptans, so I probably have to go with other medications. Patients with renal failure, for instance, obviously I have to limit the use of the NSAID or try to avoid NSAID. So keep all of this in mind. That will take us through the algorithm of which drug we choose, remembering that, again, migraine is highly comorbid with many other conditions. Psychological conditions, psychiatric conditions, cardiovascular factors, asthma is far more common in patients with migraine as compared to the general population. So you have to keep all these elements in mind before you choose your treatment. Now, what comes down to preventive therapy, if that was the second arm of the question, again, I will go through the same algorithm, the patient's characteristics, profile, comorbidity factors, before I would choose the medications. Assuming the question indicates that we have no access to the GPANs or the injectable CGRPs, and that was the case, as a matter of fact, more than 5 years ago. At that time, we had no accessibility to any of the newer migraine-specific therapy. So what we used to do at that time, well, obviously, again, we go through the same process, looking at the patient's profile and looking at the patient's characteristic and try to choose a drug that can cover some of the elements that patients would have. For instance, the patients have insomnia as one of his problems. I would choose a drug such as amitriptyline that can obviously tackle insomnia, as well as can be a good option for migraine prophylaxis. So a patient, for instance, had epilepsy, which, we just had heard that there's some genetic overlap between epilepsy and migraine. So you probably choose a drug that can tackle both of these disorders, such as topiramate or valproic acid. So all these elements have to be taken into consideration before you choose your medication.
And I have a lot of questions around menstrual and hormonal migraine. I don't know, how often do you see this in your practice, and do you treat them different?
Well, obviously, menstrual migraine is one of the big problems we see with women with migraine in general. When we have patients like the one that I have showed you, where she had only migraine around her menstrual cycle, usually a few days before or a few days within her cycle. We had also patients that had migraine worsened during the menstrual cycle, but she also had migraine attacks outside her menstrual cycle. So we have two types of patients with attacks only during menstrual cycle, and patients with attacks within or during the cycle outside, as well as outside her cycle. Again, How you tackle this patient, again, we have to differentiate. Are we talking about the CGRP era or before the CGRP era? Now, after the CGRP era, I think we have the availability of the monoclonal antibodies, which we have been using, though there is no data yet on the use of monoclonal antibodies in terms of the muscle-related migraines. Similarly, there's no data on the situational migraine with GPANs, though I've shown you data that it works, because I believe there's a role on rimegepant or the GPANs in those, what we call it, situational migraines. There's some anecdotal report similar to the cases the case I've shown you that they do work. But aside from this, it's had to be, again, quite individualized. There's recommendations in the past by the American Headache Society recommending some of the triptans, such as frovatriptans as well as zolmitriptans. And in terms of muscle-related migraine, I have extensively used them. But the problem is, if they fail, what next? Again, if they fail, we have now options. You can certainly go anecdotally, off-label, the use of rimegepant if you have an access to this. Monoclonal antibodies can be used, again, this off-label, because there's no data to support their effectiveness in that form of therapy. But if patients had both, meaning menstrual migraines as well as migraine outside the cycle, obviously monoclonal antibody, if available, can be used. If unfortunately you don't have access to this medication, then you can try any of the other previously available, what we used to call them standard of care therapy, though I personally don't believe that
yeah, me too. I'm not happy with the standard.
But that's what we are using now in the literature. When we make the comparison between CGRPs and oral therapy, we call them standard of care. Are they standard of care? I believe not, but this is what they call them.
I think they were the only things we had. So they're standard only because of lack of other
available options.
Yes. Available options for us. Then the question is around combination. Do you combine in your experience, do you combine? Like, one of the things we see very often in people living with migraine is the nausea. Do you use an antiemetic at the same time? Do you use another, like if a GPANT or a monoclonal does work, but not well enough, do you add an NSAID? Do you add an antiemetic? Do you have any experience of combining these medications?
Obviously, we are practicing an evidence-based era. And everything we do, we have either look at the evidence base or we have to rely on our experiences or anecdotal reports. Now, again, if you differentiate acute therapies versus preventive therapy. Now, for preventive therapy, obviously, not everyone is a responder to CGRPs, and that we have certainly non-CGRP respondents. And I totally agree with the previous speaker. And I believe that at some point in time, maybe with the help of pharmacogenomics, we should be able to identify who are these patients are CGRPers, meaning they only work, the CGRP would work perfectly for them, and others who are non-CGRP, meaning we have to tackle other receptors. Because we see in our practice now, not everybody responds to CGRP. Certainly, these patients are non-CGRP receptors. Can we identify those early before starting treatment? We don't, but I hope that with the pharmacogenomics, other markers, probably we should, at some point in time, we should be able to identify who are responders versus who are not responders. Now, for those who may not respond to CGRP, I personally have combined with other mode of therapy, again, with oral therapies, though I hate to call it standard of care, but we can certainly try one of the oral therapies with them. If in case of chronic migraines, with or without medication overuse, there are some literature to support the combination of the monoclonal antibodies with the botulinum toxin. So it seems there is some synergistic effect with combining botulinum toxin with the CGRP. So that could be certainly an option. Now, for the acute therapies, obviously, again, same thing. Not everybody would respond to GPANs. And I've seen the data as well. I mean, obviously, if you look at the pain and freedom rate, we're talking about 30-35%. So clearly, not everybody would respond to GPANs. And for those patients who do not respond to GPANs, combining out with another drug such as NSAID can be a possibility. And I've done that as well for patients who do not respond to GPANs. If they have strong nausea or vomiting, you can always combine with an antiemetic medication. But one thing to keep in mind, though, this is very critical. I think the timing of taking the medications. Oftentimes, patients do not get to take the medication early in the course of the migraine attack. They tend to take it very late. Well, they think, well, okay, let's give it some more time. The drug would, then they will start taking the medication. I can tell you right now, there's some tons of data now suggest the earlier you take the medication, the better the outcome would be. And that data had been shown over and over and over with several studies with the triptans, and now we see it as well with gepants, that once your patient get what we call the central sensitization phase, meaning the what we call it, when non-painful stimuli become quite painful, I can assure you the likelihood of your patient responding to whatever drug, triptan, non-triptan, whatever, would become exceedingly small, less than 40%. So the rule of thumb, the message across, take it as early as possible, because that would, at least you have a better chance of responding to whatever drug, triptan or non-triptan or even NSAID, before your patient get to the central sensitization phase.
I can see many more, and I know it's a little bit late. Is it? I think Maya did have some announcements she wanted to, and everybody's saying we're not going to look at the score, so I haven't looked at the score yet. I don't know about you, Dr. Sadi. We are not looking at the score. We're very committed.
But I can tell you now, I just turned to the score because I'm a Brazil fan, by the way. If you want me, I can show it. I can share it with you. No,
don't tell me. I'm gonna rewatch the thing.
Okay.
I want to say thank you very much. Again, I can see more and more questions, and unfortunately, we cannot answer all of them tonight. It's been a pleasure, and me personally, on a personal level, I'm very happy to see you again. I know that it was a pleasure working with you years ago, and to see you again is such an honor for me to be seeing you again. Thank you for a very informative and very well prepared presentation. We really appreciate you being with us. I will hand it to Maya again. I would like to thank IVPN and Pfizer for initiating this, and on behalf of the Association of Migraine Disorders, I would like to thank everybody who has joined us tonight. Thank you very much. Thank you, Maya, and thank you, the team of IVPN, for making this possible to the whole world. Thank you very much.
Thank you, Hiba. So first of all, I would like to thank AMD for endorsing our activity, and also Dr. Hiba. Without your effort, this could have not been done. So, and thank you for the excellent moderation. Also, let me thank our esteemed speaker, Dr. Leen and Dr. Saadi. Thank you, Pfizer, for sponsoring this activity, and the big thank you goes for you, our attendees, for joining us tonight despite the big football game, and for staying with us until the end. I just would like to remind you that you will receive your certificate of attendance by email within 1 week from today. And if you missed any part of this webinar or would like to revisit the lectures, you can check our IVPN NeuroPsychiatry library, where we will post all the recordings. Also, follow us on our social media platforms to know about all our educational activities, and stay tuned for more migraine master classes. We actually have prepared with Hiba 6 master classes until June 2023. Our second migraine master class is scheduled on January 30, 2022, and will discuss migraine triggers and lifestyle recommendation. Until we meet again, stay safe and have a good night.
Have a good night. Bye.
Good night. Bye.